Hello, everybody. This is our video. Um, in this presentation, we're going to discuss figures 1G to 1M in the neuroscience research done by, and I hope I say this right, Whitang, Alexei Conby, Michael Kincher, and Rolf Schneckenberger. The title of their research article is a VTA to basal amygdala dopamine projection contributes to signal salient somatosensory events during fear learning. My name is Tycho Bosch.
My name is Caden Muderis.
My name is Talar Nigerian.
And I'm Zane Tayez.
All right, so on this slide, we have a brief summary of figure 1A to 1F. In this part of the research, they had a few methods, or their approach that they used was they injected dopamine transporter mice, or the DAT mice, with an AAV1 vector. And this is how they traced the neurons in their brains. And this vector was injected in the VTA. They created images with microscopy, and they found out that a high percentage of the VTA dopamine neurons were also tyrosine hydroxylase plus neurons. So this means that these neurons were actually making dopamine. Also, what they found out was that most of these neurons were connected to the basal and the central amygdala. And what you see here in the slide is the takeaways, so the conclusions, so kind of what I said, of virtually all of the neurons, so about 80% in the ventral tegmental area, were affected by the injections of the viral vector, demonstrating the presence of the target tyrosine hydroxylase. These tyrosine hydroxylase plus neurons were separately stained and observed with Th antibodies along with green fluorescence protein to provide merged images that demonstrate clear coincidence of DAT expression in neurons going from the VTA to the amygdala. And that is what you see here in the images on this slide. In 1D, you see a good picture of the EGFP and the TH, and on the right side you see them combined, and you see that they overlap in many places. Okay, next slide. So in this presentation, we're going to discuss these figures. The title is a dopaminergic afferent projection from the VTA to different amygdala subnuclei. And the question or objective that they're trying to answer in this part of their research is, what is the location of the basal amygdala projecting dopamine neurons in the VTA?
So the approach for figures 1G through 1M is to showcase the injection of a retrograde tracer in the basal amygdala of the DAT mice. So it is an injection of CTB Alexa 647, also known as cholera toxin subunit B, which attaches to cells by binding to the ganglioside GM1, and then it produces a magenta color. And so additionally, there will be a localization of the neurons labeled by this retrograde tracer, and then they will be plotted on histograms based off of their positions within the basal amygdala at 3 different bregma levels. And then in figure 1G, this is a schematic of this retrograde labeling technique. And this is pretty much here to exemplify the different areas / locations of these dopaminergic projections into the basal amygdala, and to show how this tracer is going to be working and what it looks like when things have been traced. And then so here in figure 1H, we see the 3 different bregma levels. So we have negative 1.34 mm, negative 1.46 mm, and negative 1.58 mm. And these are images of the basal amygdala at 3 different coordinate positions after the injection of the CTB Alexa 647 tracer. And these magenta spots represent where it has been traced. And the bregma is actually the points of junction where the sagittal and coronal sutures meet. And so this is where the different levels come from.
Okay, so for figure 1L, the retrogradely labeled neurons were found in the medial prefrontal cortex, or the MPFC, the hippocampus, and other brain areas known to supply input to the BA. However, this data wasn't shown specifically in these figures. A photo of the VTA was, however, taken after both the tracer and green fluorescence protein were added. Projectors with back labels are specifically designated by the tracer, and the GFP specifically identifies neuron with DAT. Figure 1J, retrogradely labeled neurons were also observed in the VTA. In this image, the tracer GFP is visible under the microscope, and the BA projecting neurons of the VTA. Confocal fluorescence microscopy was used for this observation that you can see in this figure. Confocal fluorescence microscopy is a microscopic technique that provides true three-dimensional optical resolution. In microscopy, 3D resolution is generally realized by designing the instrument so that it primarily sensitive to a specimen's response coming from an in-focus plane, or by subsequently removing the contributions from out-of-focus planes. In figure 1K, the position of neurons in the VTA containing the CTB tracer may be seen as a graph of dorsal ventral orientation. If you look at this graph or figure on the left side of the histogram, it appears that BA projectors are mainly found dorsally, with the VTA containing the CTB tracer.
All right, so figure 1L, similar to the previous slide, in figure 1K, this graph represents the number of neural projections that are deviated from the mid-sagittal line rather than deviated from ventral or dorsal. And as you can see by the data that's been plotted, it is a pretty homogeneous distribution within their boundaries, which are represented by the black dashes, implicating that there isn't really any sort of specificity to when the neurons project from the left or right side, and actually focuses more on the dorsal and ventral side. And in figure 1M, this is a graph representing the number of traced BA projectors found in another mouse from a previous study, which sort of serves as an outlier from the data that's presented before as a counter argument. And it shows for deviations in the posterior and lateral DAT projectors. As you can see, there is a slightly less number of neurons and projectors in this graph, and this is likely due to an anatomical fluctuations in the VTA volume because it differs between each specimen. So in conclusion, all the data and figures concisely conclude that there is definitely a correlation between the specific regions of the VTA and the specific regions of the amygdala, primarily the basal amygdala and the central amygdala. And these said pathways are definitely connected through neural projectors that have the dopamine transporter. And this emphasizes that the study's findings that the VTA and the amygdala are connected through dopaminergic neural pathways. Any questions? Okay. Thank you.